Hello everyone. Um, it's my third time recording because my laptop hates me. Anyways, today's video, and it is a video, it's not a meeting, is just about transmission genetics and just a general recap. I'm gonna use the presentation that the teacher used because this is what you guys learned from, and hopefully you can relate more to it. So, what does genome mean? Genome is basically all genetic material of the organism. So that's all the sequence of DNA, all your codons, everything that codes for every physical characteristic that you have in a haploid. So that means in a haploid, meaning gamete, okay, meaning half. Okay, so in your cell, you have diploid genetic material. You have 2N, okay, that means that you have two chromosomes. Okay? You have 46 chromosomes, and half of that would be 23 chromosomes. Okay, so each chromosome is made up of chromatin. And in meiosis or gamete formation, you will see that these chromosomes get separated, these chromatids get separated, and they go to opposite poles of the cell, and then you'll get half. So you'll get each chromatid being inherited independently, which we'll get into later. Anyways, what's next? Allele. What's an allele? An allele is a variation of the gene. So this here, you'll have two gene locations. So you'll have two locations of a gene. A gene is, by the way, a unit of inheritance or a sequence of DNA. Okay, you'll have two locations, location A and location A, or this is location A. Okay, you will have two versions of a gene at that location. Okay, or two alleles, big A and small a. Okay, this is allele small a. They're both in the same location, and they're both exactly the same gene, but they're different variations. So, yeah. What's recessive and what's dominant? So dominant means that if it's there, then it's always going to be expressed. Sorry guys, that was my mom bringing. Anyways, small a, big A, meaning dominant, meaning that you only need one version of it to be expressed. Okay, so here you'll have one big A. So that means it doesn't matter what's here. It's always going to be the dominant. Okay, so if say the dominant allele codes for brown hair, and the recessive allele codes for blonde hair, then you will, if you have the dominant allele, you'll always have brown hair, even if you have the allele for recessive hair as well. Okay, the recessive allele means that you need two of it in order to express the phenotype. So you'll need two small a's, two of the same version of the allele in order to express the phenotype. You also can get homozygous dominant, homozygous and heterozygous. Homozygous meaning that you have two of the same allele. Heterozygous meaning that you have one dominant and one recessive. Locus, like we said, means location or place on the DNA sequence. Genotype is what you have. Okay, so what combination of alleles in your DNA. So you'll get one on the chromatids, you'll get one dominant, and on the other chromatid, you'll get recessive. Okay, so your genotype would be big A, which is dominant, small a, which is recessive. Phenotype is your physical characteristic. So this is what you see, okay, whether it's hair color or skin color or eye color or so on. Homozygous and heterozygous, we already mentioned them. Codominance and incomplete dominance, two very important concepts. So, first of all, about let's talk about codominance. So codominance means that you have two alleles and the two of them are dominant. So you don't have dominant and recessive, you have dominant and dominant, okay? So what you will get is, let's say you have a cow, okay? A cow and a bull. The bull is black and the cow is white. both white and black are dominant, or the alleles for white and the allele for black is dominant. So if they were homozygous, and they will be, because well, let's say D is for black bull and W is for white cow. So because both alleles are dominant, you will get the genotype BW. And because they're both dominant, you will get both black and white phenotypes. So you cross a black bull and a white cow, and you will get a cow or a bull that is white and black. So you will see white patches on the bull or the cow. Incomplete dominance. So this is similar. You have two dominant alleles, okay? However, when you're heterozygous, so when you have the two alleles, so BW, it will be like an intermediate or a mix. So it's like getting white paint and black paint and putting them together, stir it a bit, you get gray paint, okay? So that's incomplete dominance, is an intermediate or mix. Polygene or polygenetic inheritance. Polygenetic inheritance refers to multiple genes at multiple locations affecting the expression of one phenotype. So for example, height. Height is affected by a lot of genes, okay? Not only that, it's also affected by the environment. So if you have the genes for height, each of them contributing very little, but it accumulates, okay? And you also have the right nutrition growing up, you will be tall, okay? Same works for BMI or size, or hair length, or I don't know, health. All these are polygenetic, and they're affected by many genes, but also the environment. Here's a further explanation. Pleiotropy. So pleiotropy is effectively the opposite of polygenetic inheritance. Pleiotropy meaning that one gene influences multiple attributes. And we have to remember that genes are responsible for coding for proteins, which carry out biological processes. And these biological processes being carried out leads to the physical characteristics that we have. A good example of pleiotropy is albinism. So albinism is the lack of melanin. However, it's not just the lack of melanin, it's the lack of melanin at a certain point. So melanin is a derivative of tyrosine, the protein tyrosine. There's an amino acid tyrosine, okay, gets processed and modified, and then it can be melanin. So what happens is because you have a lack of melanin, because you don't have the protein to turn tyrosine or to process that tyrosine into melanin. So what you will get is you'll get the lack of melanin in different parts of your body. So first of all, skin, you will be very pale, and because you're unable to produce melanin, which is bad, because you will not be able to absorb UV, and then you're at a great risk of skin cancer. The hair, your hair color would be very light, blonde, because there's no melanin to give you hair color, okay? What else? Eyes, your iris even has melanin. Well, if you're brown-eyed. So having a lack of melanin will give you either blue or green eyes, because your iris will be translucent. Lethal alleles. So lethal alleles are any alleles or a combination of them that leads to death, either at an embryo stage or a childhood stage or adulthood stage, okay? So yeah, that's lethal allele. Lethal alleles can be can be dominant or recessive. So if it's recessive, that means that you need to be homozygous and recessive in order to get the lethal, or to die. However, if you're if the lethal allele is dominant, that means that if you have the dominant version of the allele, then it will lead to death. Okay? Multiple alleles. So multiple alleles is best explained in terms of blood type. Okay, so we all know the blood types A, B, AB, and O. Okay, so A, B, AB, and O. Okay, for blood type, there is actually three. Hold on, there's three alleles. Okay, A, B, and O. So that will mean that on a chromatid, you can have either A, or you can have B, or you can have O. Like so. Okay. So, of course, on a chromosome, there are two chromatids. Okay. So you can have on one of the chromatids A and on the other B. In which case, because A and B are dominant, so they're both dominant, codominant, you will get AB, blood group AB. However, if you have a chromatid with A and a chromatid with O, your blood type will be A because A is dominant over O. O is recessive to both A and B. Okay, so if you have A O, then you have A. If you have B O, then you have B. If you have O O, then you have O. If you have A B, then you have A B. And that's exactly what that says. So a true breeding organism is an organism that is homozygous. Okay, so any of these following combinations that are in this presentation are pure breeding. A gene, like we said, is a unit of inheritance. It's a part of the DNA. Okay, you basically just splice that part. Okay, and there you go, gene. Of course, parts coding for a certain characteristic. Okay, so you'll have the gene for the eyes. That's a certain part. Anyways, or eye color. You don't need to know much about this slide. But if you're curious, introns and exons, what that means is exons are the parts that are expressed. Exon expressed. Okay, so exon is basically the end product. You will have a gene with exons in it. The introns are the parts between the exons that aren't expressed. They're not always useless, but sometimes they are. So these are spliced. Okay, so that's the processing of mRNA, and you will also gene is a part of genetic material, so it's not just DNA, it can be RNA as well. So the introns just get kind of like taken out, and you will get the exons. They will get put together. Yeah, a human genome. How tall it is? Mendel's law of inheritance. So there are, for now, two laws of inheritance. The first law is the law of segregation. And what that law says is that if you have a chromosome, okay, you're like a Black Panther. Anyway, if you have the chromosome with two chromatids, you will have, during meiosis, okay, you'll have that chromatid with an allele on it, and you'll have that chromatid with other allele on it, okay? So your gametes each contain chromatid with the allele. So if you're heterozygous here, you have big Y, small y. One of your gametes will have big Y, one of your gametes will have small y. And then if you're a male, you'll have a sperm. Your sperm will combine with the egg of the female or the mother, or if you're a female, you have an egg, of course. And then you'll produce a zygote. And a zygote it has the diploid, so it has all the genetic information. Okay, it will have an allele or a chromatid with the allele from the father, and it will have the chromatid from the allele from the chromatid that is from the mother. Okay, so the two gametes mix, and then you'll get the zygote, the individual. And then you'll have Mendel's second law, which is the law of independent assortment. And all that says is if there are two genes at different loci, different locations, okay, not only different locations, but different chromosomes, okay, they will be inherited independently. They will So they won't have to do anything with each other. They won't be linked. Okay. So let's say if I have here in the example, if I have two heterozygous individuals, okay, then I will the gametes will include all the combinations, okay, because they're inherited independent, they're inherited unlinked to each other. So for let's say, look at this cat, first cat, the brown cat here, the one with the purple background, the one that is heterozygous. Its gametes will be big S, small b, and then big S, big B. And you'll have the other gametes. Well, that's two gametes, and then you'll have also small s, small b, small s, big B. Okay. All right. So that's that. These are some symbols that you might need in a bit. Sorry, I am in my pajamas and yeah, I'm not dressed as smartly as I am on the Zoom meetings, but I'm at home. There's no reason to dress smartly. Much better. Anyways, crosses. So crosses, like we said, I've already been doing them, and all of these are the combination of gametes. Okay, so you get big A, small a. These two are the gametes, and then you combine them to form a zygote. Okay, dihybrid cross is, like we said, this is exactly like we're talking about. Okay, so you will have, because the two genes are inherited independently, you will have all the combinations. Okay, so if an organism has the genotype, the big A, small a, big B, small b, yeah, then the gametes would be all the combinations. Okay, so it could be A, all the combinations of A and B. So it could be big A, big B, and then small a, big A, small b, and small a, big B, and then small b, like that. Okay, so these are all the gametes, and then you cross these with the other parent. Let's say, for the sake of simplifying, that the other parent is also heterozygous, so they will have the same combination of gametes. Okay, then you cross them. Okay, so the A, big A, you have A, both big. It'll be like this. That's how you do a cross. Number of gametes. So the number of gametes, okay, because it will sometimes it might ask you about the number of gametes, is 2 to the power of n. N being the number of heterozygotes. So let's say an organism has a genotype, big A, small a. Then the number of gametes would be 2 to the power of n. N is the number of heterozygotes. Heterozygous means one allele of each. So A and A, that they're different. That's one heterozygous. So 2 to the power of 1 is 2. And then you'll have the second example. You have two heterozygous. Big A, small a, and then big B, small b. I need to call someone to get me my water. Right. Okay, so for this third example, you have big A, big A. That's not heterozygous. That's homozygous. Okay. And then you have big B, small b. Okay, which is heterozygous. So 2 to the power of n, that would be 2 to the power of 1, which is 2. بقول لك ايه، هات لي الشحن بتاع اللابتوب بتاعي من الشنطه. عارف الشنطه بتاعتي فين؟ حبيبي شكرا. So my brother should be walking in with the charger anytime now. And then for the third example, sorry, for the fourth example, you have a homozygous, AA, and then big B, small b. That's one heterozygous. And then another heterozygous, big C, small c. And then another homozygous, and then another heterozygous. So you have 3 heterozygous in total, which is 2 to the power of 3. So for the number of gametes, 2 to the power of 3 is 8. 2 times 2 times 2 is 8. So exercise number 1. We have homozygous black short hair. So that means, okay, we have 2 phenotypes. So we have 2 alleles, okay? Well, 2 genes with alleles. Anyways, and then you have brown, long-haired Dachshunds like this, the sausage dog. They look very funny. It is known that the black coat is dominant over the brown coat, okay? And the short coat is dominant over long. And it says the first dog is homozygous. So that means that they're homozygous dominant because black is dominant, and short is dominant, short hair is dominant. So that means that they will have the genotype for the first parent. So we will have the parental genotype. So for the first parent, it will be big B because it's black, and then short haired. Say S, okay, big S. So the genotype for first parent would be big B, big S. And then for the second parent, we know. Now here's the thing. It says that it's brown and long haired. Okay, it doesn't say whether it's homozygous or heterozygous. So how do we know? We know by the fact that it says that brown long is recessive. So if it's recessive, then it needs 2 of the same allele to be expressed. So we know that it's homozygous because it says it has brown and long hair. Brown and long hair is recessive. So it will be recessive at both, homozygous recessive, both. Okay. So the first question, parental genotypes, is already done for us. We just have to figure it out. Like so. These are the parental genotypes. And then genotypes and phenotypes of F1. Well, if we're crossing 2 homozygous, by the way, do this in your own time. Cross 2 homozygous. You'll have a gamete. You'll have here. I wonder if I can show it to you guys. So you'll have ES, not sorry. Then you have small e, small b. Like so. Okay. And if you cross them, you will find that you will always get the phenotype, the genotype, small big F, small f, like so. Okay, you'll always get that genotype. So we already know the genotype of F1. And we know that black and short hair is dominant over brown and long haired. So we can say confidence that the phenotype of F1 is going to be black and short haired because they're heterozygous, and the big B, coding for black hair, is dominant over small b, coding for brown hair. Okay. So that's exercise 1 done. Easy. Farmer crossed brown haired rabbit with short ears and the red haired rabbit long ears. It says in the F1 generation, he received only brown haired with short ears. So we know that brown haired with short ears is dominant. Why? Because it says here that it crosses brown haired rabbit with short ears and a red haired rabbit with long ears. Okay. So if the brown haired rabbit with short ears was heterozygous, then what you would see is you would get brown haired rabbits with short ears and red haired rabbits with long ear in the F1 generation. But it says that we only get they all have the same genotype. And when does that happen? When they're heterozygous. And when do we produce heterozygous? All of them heterozygous. When we cross two homozygous, like we did in the last question. So it is known that these attributes are inherited independently, give the genotypes of the parents and the F1 generation. We already know what it is. We know that both parents are pure breeding and the generation is heterozygous. Okay, I hope that makes sense. I know I haven't explained a whole lot, but it's just deduction. Okay, so here I'll try to explain it. Brown-haired rabbit with short ears and red-haired rabbit with long ears. Okay, in the F1, only brown-haired. The only case where this happens, okay, is when they all have the same phenotype, is when they're all heterozygous. Okay, so if they're all heterozygous and they have that phenotype, that means that their genotype is the dominant one. Okay, so that means that brown hair and short ears is dominant, red hair, long ears. So the genotypes of the parents are going to be as following. And the F1 generation are all going to be heterozygous. Parents, that's the first generation. The phenotypes and genotypes of the F2 generation at the crossing F1. So this is actually quite tricky. The F2 generation, F2 is the grandparents, grandchildren generation. But it says if you cross one of the F1, well, we know all of the F1 are heterozygous, okay, with a double recessive homozygous. So with a red-haired, long ears. So that would be effectively doing this. Okay, that's the F1 generation. They're all heterozygous. Okay, and then that's the homozygous recessive, double homozygous recessive. Okay, which is the red-haired, short, long ears. Okay, so we can cross these together. I'll do it. Shouldn't take a lot of time. So we get 4 genotypes. Like so. Okay, that's a dihybrid cross. Okay. Yeah. Combination of these and combination. You get 4 genotypes. Okay, I made a mistake here. That's supposed to be big S, small s. Okay. And then there we go. After genotypes. And the phenotypes would be, well, we have the dominant and we have the recessive. Okay, so you will have brown-haired, short ears, and you will have brown-haired, long ears. You will have red-haired, short ears, and red-haired, long ears. So everything, basically. Okay, so this question has a bit of a twist. It says a woman with blood type A, with A blood type, has a hearing disorder, while a man with AB blood group has normal hearing. But he is a carrier of the defect. Okay, now I'm going to explain what that means, but bear with me just a second. They have a child with blood type B. Okay, and then it asks us for the parental genotypes of the blood group. So the woman has blood type A, and we said that A is dominant over O, but is co-dominant with B. Okay, so if she had, there's only two cases that she can have a phenotype blood type A. Okay, is if she had the genotype AA or AO. Okay, the man has the genotype AB because he has the phenotype AB and they're both co-dominant. Okay, so that means that, and then when they have a child, the child has blood type B. This can only happen if the woman has genotype AO. Why? Because if she had the blood type or the genotype of the blood group AA, then the cross would be like so. Okay? None of the offspring have blood group B. That means that the woman has blood group or blood genotype AO, because if she had AA, then she could never have a child with blood type B. So we know that the genotype of the mother is already AO, and the phenotype of the father is AB, meaning that he has both alleles, A and B. So we know that his genotype is straight away AB. That was nice. Okay. So now we know the parental genotypes. To answer the first question. Genotype and phenotype of F1. F1 is a child. So his genotype would be BO, because we know his mother is AO and his father is AB. And he has a blood type B, so he must be BO. Okay. And the phenotype is blood type B, which they've already given to us. Okay. And then it asks about the probability that the child with blood type B will have a hearing disorder. Now, here's the part that I told you to bear with me just a second. The woman has a hearing disorder, but the man is a carrier. What does carrier mean? Carrier meaning that he is heterozygous. His offspring can have the disease, but he does not. Okay. So the only case where this works is if the disease is recessive. Okay. So if he's a carrier, then we know straight away he's recessive. Okay. And this man is heterozygous. The disease is recessive. And the mother has the disease. That means that she has a homozygous recessive, because recessive is only expressed when it's in the homozygous state. We know that when we're crossing, it's going to be, let's say, hearing H. Okay, so the man is heterozygous, so it's going to be H, a big H, small h, and the woman is homozygous recessive because she suffers from disease. So the cross is going to be as following. There we go. No, mine is heterozygous. So you will get 50% chance that the child will have normal hearing and 50% chance that he will have affected hearing. If that makes sense. A farmer discovers an oddity among. I'm just checking that I'm recording because I may have forgotten. Anyways, exercise number 4. We have been asked to exercise by someone. A farmer discovers an oddity among his sheep. Some of the sheep have abnormal large eyes and both legs. Crosses of these sheep. So he had 2 sheep with abnormally large eyes, and both legs, and he crossed them together. That bad idea. Anyways, and 2/3 of the offspring have a normal phenotype, but one third. Wait, no, 2/3. One third is normal. So they're okay. So how can we explain this? Okay, so there are two. This is a disease, okay? And there are two ways or recessive, okay? If it was recessive, that means only express when it's in the homozygous state, okay? If it's in the homozygous state, then both parents would have genotypes homozygous recessive. So they will always produce homozygous recessive. Okay? That means uh, in this case, large eyes and broad legs is not recessive, it's dominant. Okay? Because you get one third that have the normal phenotype. So that means that both parents are heterozygous. Okay? Because if they were both homozygous, uh, and the disease was recessive, that means that all offspring would have the disease because they will cross it. So if I cross it like this, the disease is small a, for example. Okay. Will always produce this. But if you are crossing two heterozygotes, then there is a 1/4 chance that the offspring doesn't have the disease. Here it's 1/3, but it's close enough, so it's okay. So yeah, now we can tell what's the explanation for this type of inheritance that appears here. We can tell that this is dominant inheritance. So the allele causing the abnormally large eyes and both legs, it's a dominant allele. Exercise number 5. Pale colored horses are crossed with chestnut colored horses to produce palomino, intermediate coat color. Intermediate straight away. If you mix it, making an intermediate, that means that it's incomplete dominance. I already know the answer to the first question. Okay, so we should know that just by reading the first sentence, intermediate coat color, straight away incomplete dominance. Farmer have the English here is not best. Farmer has 2 palomino horses. Okay. And the number of mating between them. So he has 2 palomino. Well, we know it's incomplete dominance, and if they have an intermediate color, that means that they must have a heterozygous phenotype because they have an intermediate. They must have dominant alleles. Okay, so let's say the allele for pale color is what? P, big P. Okay, the parents will have big P, and then the other parents, which is chestnut C, also dominant because it's incomplete dominance, so they're both dominant, okay, and they produce intermediate. And then the offspring would have genotype PC. Okay, so now we know that what answer the question: what type of expression is suggested? It is incomplete dominance. What is the parental genotypes? Well, it's going to be big P and then like that. Okay. And then genotype of F1, which is the intermediate or the palomino, is going to be heterozygous state. Because like this. Okay. Oh, that makes sense. Epistasis. Everyone's nightmares. So there are 5 types of epistasis: recessive, dominant, duplicate recessive, duplicate dominant, and dominant and recessive. Okay. So what does epistasis mean? Epistasis means that one gene, certain state, dominant or recessive, will affect the expression of another gene. Okay. So I want to explain this in a biological way. Okay. Let's say you have a mouse or a mouse, okay? And in order to produce or to color this mouse depends on 2 processes. Process 1, and process 2. Okay. So this is the first. Okay. This is process 1. This is process 2. Okay. This is the end product. So here I'm going to use recessive epistasis as an example. If we have recessive epistasis, okay, that means that for process 1, we need a certain protein, okay? And process 1 is responsible for producing pigments. So process 1 says whether you have pigment or not, okay? Or whether you're albino or very white, a very white mouth. Then process 2 says whether it's white or black, sorry, yellow or black, okay? So if process 1, if something happens with process 1, doesn't matter what happens at process 2 because there's no pigment produced, okay? So you're always going to be white, no matter of what color is coded for, okay? There's always going to be white. So there's a certain protein here, and in order to correctly code for this protein, you need a dominant allele, okay? If you have 2 recessive, then you're not going to have this protein, okay? So if you have 2 recessive, you're always going to be white, okay? 2 recessive at this point. So 2 recessive of the gene that produces the protein that carries out process 1. However, if you have anything but recessive, and then here you have a dominant, okay, then you will produce, let's say, yellow. Okay, if you have 2 recessive, then you would use black, given that there is a dominant allele here. There isn't a dominant allele here. Doesn't matter what's there, because you're not going to have pigments. You're always going to be white. Okay. So that being said, yeah, here's the example: white, black, and yellow. Okay, so here we can see that there are 3 phenotypes. Keep this in mind. This is going to be important. 3 phenotypes. Okay, you have 4 white mice. All the white mice are homozygous recessive at the C. Okay, so they have 2 small C's at the CG. That means that their process 1 isn't going. Okay, so they're always going to be white. It doesn't matter what the A is. Okay, if it's dominant, it doesn't matter. If it's recessive, it doesn't matter. If it's they're heterozygous, it doesn't matter because C is recessive. That means it's always going to be white. However, if it's non-homozygous recessive, then your color depends on the allele in the first locus, first, which is A. Okay, so if you have big A, it will always be yellow. If you have 2 small A's, it will always be black. Well, unless you have 2 small C's, in which case you're going to be white. So that's recessive epistasis. Okay, it produces a ratio of 9 to 43. Don't believe me? 7, 8, 9, 9, 1, 2, 3, 4. So 9 to 43. Okay. Now, duplicate recessive epistasis. So what this means is it works both ways. Okay. So if you have, hold on, I have another picture. Yeah, there we go. So in this example, for let's say here in this flower, okay, have 2 genes. Small p, well, have gene P and gene C. Okay. In duplicate recessive, if you have homozygous recessive at C, you're going to be white. You're going to be a white flower. If you have homozygous recessive at B, you're also going to be white, or white flower. Okay, you're going to get white. The only case when you don't get white is when you have heterozygous, in which case you're going to get purple. Okay, so that's how that works. Okay, if they're epistatic to each other, in the homozygous recessive state. Okay. So notice that here there's only two phenotypes. Two phenotypes. Whereas before there was three phenotypes. In recessive epistasis, there's three phenotypes. Duplicate recessive, two phenotypes. Dominant, three phenotypes. Duplicate dominant, two phenotypes. Dominant and recessive, two phenotypes as well. So, dominant epistasis. So what this means is the same scenario. Well, if you have a dominant allele at a certain locus, so let's say, in this case, it's A, okay, because it's an inhibitor. If you have a dominant A, okay, and it doesn't matter what B is, you're never going to produce pigment, okay? Or it's going to be a white pumpkin. We're talking about pumpkin fruit. So it is that time of year, isn't it? So yeah, you'll get, if you get a dominant allele, big A, then you'll always get no pigment. However, if you have homozygous recessive at A, then your color depends on B. Okay, if you have dominant B, then you're gonna have yellow. If you have small B, then you're gonna have orange. Okay. So yeah, you'll have three phenotypes. Okay, if you have dominant A, if you have dominant allele at A, then it's always going to be white. Okay. However, if you have homozygous at A, dominant at B, then then you're going to get yellow. You have homozygous recessive at A and homozygous recessive at B, then you're going to get orange. Here, notice again, 12 to 3 to 1, that's the ratio. 3. Okay, so duplicate dominant. Here, it works the same way as duplicate recessive, but it's dominant. So if you have dominant to A, no pigmentation. If you have dominant to B, no pigmentation. The only case when you get pigmentation is homozygous recessive at A and homozygous recessive at B. Okay? So dominant and recessive epistasis. So let's say if you have dominant to A, okay, dominant heterozygous for A, then you're not going to get any pigments. Now we're talking about onions. Variation, mice and pumpkins and onions. So yeah, you guys hear onions. Anyways, you have dominant A. Okay, doesn't matter what else you have, you're going to be white. If you have recessive homozygous at B, doesn't matter what you have, you're also going to be white. Okay, so the only case when you cannot be white or no pigmentation is when you have homozygous at A, homozygous recessive at A, and dominant homozygous or dominant heterozygous at B. Exercises, last point, and then we'll go to maternal, but that's not much. And you have Haytham's video. Okay, so in a culture of crocuses, which I think is flower, pure lines with white flowers and purple flowers are carried out. As a result of the crossing, the representatives of the two purebred obtained in the, so first of all, it says in the F1 generation, there's only white crocuses, okay? Interesting. F2, now here's where it gets interesting. In the F2 generation, so when you cross two of the F1, you get white, purple, and yellow. Now, how many three. Now, no, eliminate duplicate recessive, eliminate, and we can eliminate recessive because these produce only two phenotypes. However, dominant and recessive epistasis, dominant epistasis and recessive epistasis produce two phenotypes. Okay, so it's one of these. So as a result of crossing the representative of the two purebred obtained, so we have two purebred. We can write that cross down. Page. We'll have what's it called? White and purple. We'll have W. And we'll have hold on. Because they're purebred, okay. And then in the F1 generation, all are white, so they're all heterozygous because we're crossing two purebreds, where two different purebreds, okay, one dominant and one recessive for two different colors, okay. So you will always get heterozygous. A bit slow now because it's starting to get late. Okay, so that's the phenotype of F1. Okay, now it says that all of these are white, and we know it's either dominant or recessive stasis. All of the heterozygotes are white, and they're heterozygotes. So that means it must be dominant because there's no homozygous recessive in the offspring, and they're all white. Okay, so that means the only case when you can get purple is we can get homozygous recessive at both loci. So that means at both genes. So that means that the mechanism is dominant epistasis with the white flower, dominant epistatic, the purple flower. Okay. So, first of all, genotypes of the parents. We know that they're both two purebreds, so and we already given them. Okay, and then in the F1, going to be this. Well, that's F1. Okay, and then they're going to be white. F2. Now it says white, purple, and yellow. So for the white, it's going to have dominant. So you're just going to make the cross between the heterozygotes. You're going to have two heterozygotes, and you'll find the ratio. You can give each color the genetic combination of alleles. So that's not an issue. Well, not here. Okay, exercise number 7. This one. I hate it. Anyways, in the laboratory, 2 pure albino mouses, mouse lines. Okay. Once a result was obtained, oh my God, so hard to read. In the laboratory, 2 pure albino mouse lines were obtained. Once as a result of error, mice, from the first line and second line were crossed. Oh my God, this is so hard. It's insane. Giving me a stroke. The whole offspring of the mice was gray. So, of course, no, I don't mean gray. Was gray. So F1 were all gray. While in the next generation, they had both gray and white. Gray and white. Gray and white. So that's 2 phenotypes. Straight away, eliminate dominant epistasis and recessive epistasis. Either duplicate recessive, duplicate dominant, or dominant and recessive. Now we need to figure out which one. So 2 pure albino mouse lines were obtained. Okay. As a result of error, mice from the first line and the second line were crossed. So we know they're pure breeding, but we don't know the combination. Okay? So the whole offspring of the mice were gray. That only happens in the case where all of them are the same phenotype. Is all of them are the same phenotype. Is when they all of them are heterozygous. Okay. So if they're this is an awful question. F1 is all gray. All of the F1 generation. Okay. And it says that we have two pure albino mouse lines, and we bred them. Okay. That produced all gray. All right, yeah, let me explain. I just had a breakthrough. So both gray and white mice were born. Yeah. If there are three cases, okay? And here we're breeding two pure, okay? So it's gonna be like so. Hmm. Actually, no. Hold on, just give me some time about this. Okay, so it didn't take me a lot of time. It took me a bit, but I think I figured it out. So if we're talking about two pure breeding and they're two, both of them are white, there are two scenarios. Okay, you have, and oh yeah, by the way, F1 is heterozygous because they're all the same phenotype. Okay, so it's not homozygous because you're crossing two white. Okay, and the two white are homozygous and pure breeding, but they're from different lines. Okay, and that produces gray. So what I'm trying to say is if they were both, if both mice that had been crossed had the same genotype, then you would always get the same, the same genotype and the same phenotype. You would always get the same phenotype all over again and again. So it would never produce very nice. However, it has to be duplicate, recessive, or dominant and recessive, because there are two different genotypes that produce white mice, and in that case, you could produce heterozygotes, and then heterozygous mice in the F1 generation, which are gray. Okay, so that's why I say they're heterozygous, because if both parents have the same genotype and the same phenotype, then you would never have a change in phenotype or color. Okay? So the fact that they're gray tells me that they're heterozygous straight away. Okay, so now there are two ways of having pure breeding homozygous mice. Okay, who are which are white. Okay, you can get this case for that one or that case. Okay, so here this is the first case where they're pure breeding, and this is the second case. And both of these produce for heterozygous mice. ادخل بس ما توريش نفسك. Yeah. So they will, both of these cases will produce heterozygous and gray mice. Okay, and they're all gray. Okay, so in this case, in the first case, if they're both white, that means that you will have dominant and recessive. Okay, so let's say it doesn't matter. But let's say in this case you will have dominant and recessive epistasis because dominant at W produces Y, and recessive at small p also produces Y. Okay, and in this case they're both Y because otherwise they wouldn't both be Y. Okay, and in this case you have recessive here, so that would be duplicate recessive. Okay, so it can either be dominant and recessive or duplicate recessive in order to produce heterozygotes. So now we can straight away eliminate dominant, duplicate dominant. Okay. So if this is the case, that means that when you're crossing these, just give me a second. Yeah. So that's recessive epistasis here, recessive down. Wait, so that means that okay, and then it says whole offspring was gray, and then while in the next generation goes so in the next we crossed that, and we got yeah, okay, so it's duplicate dominant. You know how I know? Because yeah, it's dominant because if it was in this case, okay, where this is dominant, where W here is dominant and small p is duplicate recessive, recessive W, recessive, homozygous recessive W, then that would be white because dominant is static. Okay, so this would be epistatic dominant, so it's not dominant and recessive. It cannot be dominant, duplicate dominant. Okay, because in that case, you wouldn't have gray mice. Okay, but if it's recessive here and here, then the only case when they would be gray is when they're heterozygous. That's it. So that question was obviously a nightmare, but we did get through it. So that's what matters. Sorry, you didn't finish. Genotypes of the parents, that would be like so. Genotypes of the parents, genotypes and phenotypes of the offspring, which is, I think, don't know what it's referring to. Offspring of F2. If it's F1, then it's going to be that. If it's F2, then it's going to be all of the combination. So it would be this. It's going to take a while. So yeah, saying the mechanism is duplicate recessive epistasis. Anyways, for the genotype and phenotype of offspring, F1 will be heterozygous. F2 will have homozygous dominant at both, homozygous recessive at both, and then heterozygous dominant, heterozygous at one, homozygous at the other, and then homozygous at the other. homozygous at the first. So yeah, you're going to get all possible combinations for F2 and the genotypes. All genotypes with homozygous recessive are going to have white color, whereas the rest gray color. Extrachromosomal inheritance. So this is a form of non-Mendelian inheritance, and it applies to two things: mitochondria, because guess what? Mitochondria do have DNA, and chloroplasts, which is why interesting fact that we can track our very first female ancestor, not our very first male ancestor, because there is, well, in terms of mitochondria, we can through DNA sequencing, you can find a sequence that is shared in all the mitochondria of human beings, and that all mitochondria, your mitochondria mother, because your mitochondria, the mitochondria of your cell, your original cell, when you're a zygote, so when you're just a bunch of cells, comes from the egg. Okay, whereas only the DNA comes from sperm. So we can trace our lineage back to something called mitochondrial Eve, the very first female human being to have that sequence of DNA, and then we'll have that sequence from her. So yeah, interesting fact. Anyways, inherited only from the mother. Like I said, sperm cell contain 50 to 100, but they do not enter the egg during fertilization. Okay, but the ovum, the egg, has 100,000 mitochondria. So yeah, like I said, the human mitochondria contains 5 to 10 identical DNA. So they do have identical DNA molecules. Okay, each is made up of, so one has 37 genes, encode for 2 different rRNA molecules, 22 different transfer RNA molecules. So ribosomal RNA transfer the RNA. So you also have ribosomes in the mitochondria, or pieces of DNA that code for ribosomes, which is because mitochondria, which are organelles in our cells, used to be independent living organisms. But then somewhere along the way, they got involved. They developed a symbiotic relationship with cells, why everything that moves has mitochondria. And yeah, we have a mitochondrial genome, and any disorder in the mitochondrial DNA that causes energy production to be compromised is inherited from the mother all the time, because you don't inherit mitochondria from the father. Okay, which is why some diseases are sex-linked. Mitochondrial disease is a group of disorder caused by dysfunctional mitochondria, the organelle that generates energy for the cell. So yeah, you will get the parts of the brain. Yeah. So here you get some people have different types of mitochondria. Okay, and the ratio of normal to abnormal mitochondria is what dictates how much you suffer from the disease, or how severe the symptoms are. Okay, and yeah, if you can get little enough mutant for um, this um, dysfunctioning mitochondria, then you have to, because you don't have as many dysfunctioning mitochondria. But if you pass a certain threshold of dysfunctional mitochondria, then you will suffer from mitochondrial diseases, muscles, and these are all examples of mitochondrial disease. I'm not going to go over them. And the genotype of its mother. Maternal effects. All types of mitochondrial inheritance are example of maternal. which is inheritance on part of the mother. Like Michelle. I think that's it for today. For me, I wonder how long this meeting is. Hopefully not too long. Yeah, this part I'm going to leave to Haytham because my brain is fried. And it's not that. It doesn't require much, just knowledge and knowing like step by step how mitosis and meiosis work. These are all the genetic information, the mitosis function. That means this is also covered very well in the book, and you will get questions from the book, believe me. We'll go over all the questions tomorrow, and we may even go over this tomorrow, but that's it for today. I apologize for my attire and like how this the room looks. Yeah, that's it for today. See you guys tomorrow.